And the topic I'm going to talk about today is basically a sort of general overview and introduction to motor and social neurophysiology that we have been doing in Parma since several years, since the first experiments done by Giacomo Rizzolatti at the beginning of the 80s, in which they described many class of cells in the premotor cortex and contributed to elucidate the way in which the primates, brain in general, control movement and how the motor cortex can provide function that go beyond the typical motor function and motor planning. So just to start and to frame the topic I'm going to talk about, I would like to start with the very general questions of the kind of question that typical neuroscientists start to ask at the end of their career. And this is a question that has been asked by Rodolfo Linas, and to try to give an answer, he went back to very simple creature like this one, which is the Ascidia. The Ascidia is an invertebrate animal that at the beginning of his life cycle has a sort of head with a very primitive sense organ, sensitive to light, with a tail that enables it to move in the sea. And it moves in the sea until it finds a rock, a substrate to which to anchor. And from this moment, it starts a new phase in its life cycle in which it will never move again. And since it touched the substrate, it starts to digest its own brain. So from this example, Linas concluded that a nervous system is only necessary for multicellular creatures that can orchestrate and express active movement, which in other words means that brains serve for moving. So this is just to say that how the brains control our movement is a very fundamental question that in a certain sense can be actually identified with the function of the brain. And today I will try to trace a brief history of the discoveries and concepts derived from physiological studies of the primate motor system, which started from, I will show you, an approach which can be called localizationism, so in which scientists tend to locate in different parts of the brain specific function and motor function using the concept of motor representation, which in the last 10 years or so has been shifted more and more toward the concept of neural dynamics. So there has been a sort of overcoming of the concept of motor representation to look more closely to the dynamics that larger brain regions perform in order to control and plan movement. All this to say that any given brain region can perform a multiplicity of functions that can emerge depending on how you look at the way in which neural activity mediates behavior. And understanding this very fundamental relationship is crucial in order to develop new technology and exploit them for application in both basic and translational research, which is the last part of the talk I would like to show you. So let's start from localizationism. That a part of the brain, subserved motor function, was already known since the 1870s with the works of Frisch and Ditzig, in which they show, using first the dog and then the monkey as an animal model, that a part of the brain is motor and another one is not motor. And they simply said that the motor part is more in front rather than the non-motor part, which is in the caudal part of the brain. From this very basic statement, the research progressed by localizing more and more specifically motor function in different brain areas. This is the macaque brains in a lateral and mesial view, in which you can see that there is a cortical mosaic of area with specific connection, with specific cytoarchitectonic features and functional properties. This work has been done in most part in Parma and in many other laboratories all along the 80s and 90s, and has led to a parcellation of the brain in which each area is characterized firstly by its architectonic features. Here you can see a section of a monkey brain in which the arrows indicate the transition between the lamination, the density, the size of specific cells. Here you can see, for example, the big pyramidal cells of the primary motor cortex that send their axons to the spinal cord and have a primary role in controlling movement. And these cells stops when you move from F1 to F4, where they start again, and there is a change in the lamination of the more superficial layers. So with all these features you can set some borders, which are of course not so sharp, but they define a continuum in which distinct areas can be identified, and this can serve as a template to inject neural tracer and to characterize the connectivity of the specific brain region. At the same time, neurophysiological studies can look at how distinct brain regions, with their typical connectivity, can subserve specific function by looking at single neurons' properties. Single neuron properties can be investigated essentially in two ways: by using the extracellular recording in awake behaving animals that I will show you next, but also initially by moving from the superficial stimulation that neurosurgeons like Penfield, for example, use to map the function over the cortical surface, but applying it with a much more sophisticated technique, which is intracortical microstimulation. So using a microelectrode of a few microns in the tip that can deliver a very precise and localized electrical stimulation, so activating artificially a very small set of neurons that produce a behavior. A behavior that in this case can be a movement of a peripheral body part, such as the hand, the lips, the mouth, and so on. And in this way, they have been able to trace somatotopic representation of the contralateral part of the body all along the central sulcus of the primary motor cortex, which, as you can see in this picture, is essentially conserved across mammals, from placental mammals to primate and even human. Here is the typical homunculus, which is what we can observe if we try to represent our body surface, our muscle in this case, since this is a motor homunculus, based on the extent of the cortical surface which is destined to the representation of specific body part. And this is due to the fact that each body part has a larger representation on the cortical surface, which is proportional to the kind of precision and degree of motor control that we can exert on this specific body part. This is the typical way in which the concept of model representation is proposed. So a model representation is a set of neurons or a part of the brain that can actually represent, encode, or plan a specific movement or motor action. And this is even more evident if you use a microelectrode to capture the single unit activity of an individual isolated neurons in terms of its action potential. So during a given motor task, during an action, during a movement, a neuron can fire, and this firing activity, if you imagine, for example, that this is a pyramidal cells of the primary motor cortex, can reach the spinal cord and activate muscles through the synapse and then produce the muscle contraction. If this is the case, if this is what the neuron represents, every time you have a movement, you should expect to have an increase in the firing rate of these neurons. This is not what actually occurs in the premotor cortex. This is more similar, but not necessarily what happens in the primary motor cortex, which exert actually direct control over specific movement. But in the premotor cortex, as I will show you in a few minutes, things works in a pretty different way. So what you will see is a monkey that will perform a motor task, so for example grasping a piece of food with its hand, and you will hear the discharge of a single isolated neurons in terms of shots. We also say that sorry. We also say that the neuron fire because the activity of a single isolated neurons is perceived like a sort of a shot of a gun. And so each time you will hear a shot, this is the neuron that generates an isolated action potential. I try to bring your attention on the fact that if you look at the hand of the monkey, you will see that the monkey is moving the hand in many ways for many purposes. It will perform a grasp with a plier that will force it to invert the sequence of activation of the muscle. So if you pay attention to the hand and to the muscle activation, you will see that there are different kinds of movement, but the firing rate of the neuron will increase exclusively when the monkey takes possession of the food with its hand. So what we have proposed, what they have proposed with this pioneeristic study is that the kind of model representation which is encoded by premotor neuron is not that of a general movement in terms of flexion or extension of the fingers, but it's actually the encoding of a motor goal, like in this case, for example, grasping. Sorry. We need probably the connection with some audio. I don't know if you can hear otherwise. Here you see that the monkey is moving the hand, like here, but the discharge is just, look at the plier, here is moving the plier, but there is no discharge. So the discharge is only present when the fingers of the plier or the fingers of the monkey are closed on the food, demonstrating that actually a typical features of premotor neuron is that of encoding motor goal. From motor goal of its own, of the monkey, they move to the study of the encoding of motor goal of another agent. And this is the typical property of the mirror neurons of the premotor cortex, which fire not only when the monkey actively performs a motor act, like grasping, but also when it observes an experimenter doing the same act. This is a bit more high, the volume. This is the motor response when the monkey actively grasps. You see that the monkey moves the hand for different purposes, but there is no discharge. And this is the mirror response. Of course, this is a very old video, and it's dated back more than 20 years ago, but now these kind of properties has been demonstrated and replicated in many different studies, in many different labs all over the world, and it is very well-established evidence that the neurons in the premotor cortex, also through indirect methodology like fMRI, transcranial magnetic stimulation in humans, actually show the properties of encoding a model representation, so what is can be called grasping or biting or licking, because this kind of effect is not limited to the end, but it's spread to any body effectors that can actually perform a goal-directed action. And these kind of neural representation are recruited also when we observe another performing a similar goal-directed action. So, the way in which these discoveries were framed since the beginning took inspiration from a very famous psychologist, which is James Gibson, who said that the visual world has the appearance of inviting many kinds of behavior. This is basically the definition of what is called in cognitive neuroscience affordance. The affordance is what an object suggests a potential agent to do. So if I see this glass, the affordance of the glass is to be grasped in this way if I want to move it, or for example in this way if I want to drink it. Depending on the context and on my motor intention, what I want to do with the object, my brain needs to select specific representation in order to interact with the object appropriately, depending on what I want to do next. And this idea is that when you try to convert it in neurological terms, the brain does not simply represent movement in order to move the body, but it can exploit its own model representation to represent the world. And what parts of the world, what aspects of the world we can represent in motor terms? First of all, the space around us. So the space around us, the space which is within our reaching possibility, within the space that I can manipulate or operate in with my hand and with the reach of my arm, is encoded in motor term. And if we look at the area 4, this part in blue of the ventral premotor cortex, we can find peripersonal neuron, which are cells that instead of firing during specific movement, fire when you touch a specific part of the monkey's body, but also when objects approach this body part, like a sort of radar. So it's a contact prediction system that predicts and activates like a park sensor. You can imagine it as like a park sensor. So when an object is approaching this part of the body surface, the neuron starts firing, and it will fire until the object has touched the skin. So there is a sort of bimodal representation, which is somatosensory and visual, which serves to provide a model representation of the space around us. If you stimulate electrically this model representation, what you evoke are movement directed toward the part of the space which is represented in bimodal terms. If you inhibit by masking, for example, which is a GABA agonist, the activity of the neurons in this region, you will obtain a reduction in the readiness and amplitude of the motor reaction that the monkey displays toward, for example, threatening stimuli approaching its body part. So, motor representation can be reenacted in order to represent our surrounding space. Another most obvious features is that motor representation can be activated when we see an object. So this kind of motor representation, grasping, which I can use to grasp this glass, is automatically activated when I simply see the glass, which means that seeing the glass around us within our reach does not simply activate our visual brain region, but also activate our motor brain region in terms of neurons that encode the visual features of an object and their motor translation in the appropriate act that we should use to grasp it. And finally, there are mirror neurons that I've just shown in the previous videos, which are these cells that can allow us to exploit the model representation to encode not only our own movement, but also the movement and the action of others. So to summarize, we could say that, as the psychologist William James said in 1892, every representation of a movement awakens in some degree the actual movement which is its object. And to date, we also have a neural counterpart of this philosophical assertion. What are these for? So here you can see a scene from the very famous movie E.T., in which the boy meets for the first time with the alien, with E.T., and they start to communicate with a very spread behavior across a human and a few monkey apes species, which is imitation. Here the imitation is used for communicate to say to the other, I'm like you, this part corresponds to your body part. And this is a very direct example of what in cognitive neuroscience is called the correspondence problem, which is the question on how can I know and identify that this body part corresponds to this body part. So when you show me your finger, your hand, how can I know that this is my correspondent body part? It can be a very trivial question. You may say, well, it's obvious. It's not so obvious if you look at the children, at the newborn, that has certainly never had the experience of a face, but nonetheless, it can, since a very few hours after birth, imitate and reproduce the facial expression of the caregiver with, for example, tongue protrusion, mouth opening, and lips protrusion. You may also think that this can be an ability that is somehow innate for humans that are destined to communicate in a social way, but this has been demonstrated also in macaques. Here you can see macaques of a few hours after birth, which is, from a model point of view, much more skilled than we are at the same age. And he is extremely attracted and attentive to the experimenter's face, which is still. Of course, the time of attention is limited because it's very, very young. But as soon as the experimenter starts opening the mouth and displaying a facial gesture, the monkey immediately activates its mouth and starts to reproduce a matching gesture. This has been replicated in dozens of animals with very carefully controlled experimental methodology, using controlled stimuli, using different times of exposure, using a blank period of no imitation, and it convincingly demonstrated that also in macaques, and another study replicated this finding in apes, in chimpanzees, that there is this phenomenon of neonatal imitation that actually suggests the presence in neurobiological terms of a mechanism that could be the mirror mechanism or something else. We don't know. There are just a few EEG studies in newborns that can suggest that there are signs of the presence of such a mechanism since birth. But it is a mechanism that is extremely widespread, well beyond humans, also in apes and monkeys, and that can serve, as we can see, for example, here in apes, to learn after birth from conspecific to reproduce gestures, movement, pattern that can be an advantage from an adaptive point of view in order to survive in the environment. If we look at a more broader perspective, we can find mirror mechanism, and so the capacity of a specific part of the motor system to resonate when a subject witnesses at an action or the consequence of an action of another, not only in monkeys but also in human, where we can, of course, match motor act, complex action, and even single movement, as in the case of true imitation. And we also have evidence of the presence of mechanism with the similar features and even much more precise in birds, where we have the evidence of the much more refined and sophisticated mirror mechanism that has a role, a specific and causally demonstrated role in counter-singing and in song learning by imitation. So even a relatively stereotyped behavior like learning this space-specific song is actually based on a mechanism that took into account the motor system and its connection with the subcortical structures like basal ganglia. And these are hypotheses on which we are actively working in order to understand all the brain areas in which these kind of properties have been identified, how they work together, and how their functioning is regulated through the intervention of subcortical structures like basal ganglia. Most of this literature has been derived from single electrode and single neuron studies, which means that you insert an electrode into the brain and you can study one single neuron at a time for a relatively limited period of time. In the last 5 years, we moved to the use of much more sophisticated probes that can enable you to sample the activity all along cortical column. So from multiple recording sites, simultaneously from a different depth at an equal space. So you can have a sort of pictures of the activity in terms of single neuron, multi-unit, and local field potential of a small brain volume. In this way, we have been able to study single neuron activity for longer times, even with chronic application, which means that you implant the electrode and you leave it for days. So this gave us the possibility to investigate the activity of a single neuron in a multiplicity of tasks during observation, during object processing, during social interaction. And what we discovered is that the concept of representation should be conceived as a much more fluid entity, because there is no neuron that actually simply represent grasping or grasping for self and grasping for other, or object or space. Actually, the way in which neurons in our premotor cortex represent object in the outside world is much more articulated. And the same neuron can, depending on the context, represent my own action, the action of other, can predict the action of other based on the object which are in the surrounding environment. So the picture is much more complex. Here you can see an example of a typical motor task in the laboratory, in which the monkey is presented with an observable object and then has to grasp and lift it. It's a pretty automatic task, as you can see, so you can actually exert a very robust and strong control because you can replicate the same trial several times, but the drawback is that you are a bit far from natural behavior. So to overcome this problem, we have been using in the last years these new kinds of probe that combine the possibility to sample activity from different sites along the shaft to a 3D organization of the shaft in order to pick up the activity of a volume of cortex, of many more neurons, and to move to a neurophysiology which is much more closer to the natural environment. So avoiding to use a strictly controlled task, but picking up activity of a larger number of neurons in a relatively smaller number of trials, but in much more ecologically relevant condition. Because what we actually need and what we still lack is the understanding of the brain function in close to natural conditions. So we can control, we can isolate, we can identify specific representation, but then when we go toward much more complex approach, what the brain reveals to us is that its way of functioning is much more complex and is difficult to reduce to the extremely controlled laboratory condition in which we have been forced to work since now. So the idea is to not to abandon the control, but to move the control outside of the conventional lab in much more ecologically relevant condition. At the same time, we have been also witnessing a tendency in the literature to focus less on the concept of motor representation as a sort of physical realm of how the brain represents the outside world or our motor action, and to focus much more on the concept of neural dynamics. By approaching this problem with the idea that, as it happens in the spinal cord, for example, at the level of central pathway generator, the brains can generate rhythmic activity, and the neural population dynamic can actually be found, and have been found, even during voluntary behavior, cortically controlled, like, for example, reaching and grasping actions. Through this approach, actually we can pick up different properties. Here you can see one of these 3D arrays with the activity at the tip during the performance of a reaching grasping task. And in this way you can see that the different type of object that the monkey is going to grasp can be actually represented in a 3D space made by the first two principal components in which you can decompose the neural population activity. So this is called neural state space and can enable to represent what's going on at the level of a large population of simultaneously recorded cells and to classify what the subject is doing even based on single trial. It is a very useful approach because actually it gives up to the attempt of understanding what the single neuron is doing. So the assumption, as we have been able to see, is that the single neuron can be very noisy, and what is actually much more informative and useful also for translational purposes is to understand how the dynamics of a larger population of simultaneously activated neurons, which is what it actually happens in everyday life, do. So if we want to apply this approach to the study of reaching grasping action, an example is this very elegant demonstration of how the activity from the parietal cortex, which is sensitive to the presentation of the object, spread to area 5 when the animal prepares to move. The visual information which has been extracted by the parietal cortex are sent to the ventral premotor cortex, which ultimately lead the activation of the primary motor cortex, which is actually the part of the frontal cortex which activate muscles. By this kind of approach and by a considerable component of computational neuroscience, the effort has been that of using this kind of signal to interpret brain activity and to use these results, so to use the interpretation of brain activity by a computer algorithm to control external devices. External devices such as, in this case, a robotic arm. And in this case, the robotic arm is controlled by the activity derived from the brain of these patients, which can actually control with its brain activity the movement of the robotic arm and drink coffee in this case. But you can imagine, and there are different lines of research active in this sense, that you can use the same approach to derive brain activity from the cortex of a paralyzed patient to deliver, for example, electrical stimulation bypassing the spinal lesion, in case of spinal lesion, and reactivate the spinal motor pattern that can produce movement, or directly stimulating with the decoding of neural activity the muscles which has been deafferentated by the lesion, and in order to restore the voluntary motor behavior. So it has been very useful to conclude this part to understand that the focus, especially for translational purposes, may be that of understanding in a better way the neural dynamics that underlie specific behavior and to use them in order to restore lost function. But what we are still dealing with are neural dynamics of always essentially static system. Because in fMRI, you can enter by laying on a table and you are scanned while you are absolutely still. Movement actually are incompatible with the good signal because they produce artifacts. The same is true for EEG, which is now starting to be applied even with the wireless technology in moving person, but there are still many problems with artifacts. And the same has been true for monkey neurophysiology, in which head-fixed experiments were absolutely mandatory in order to achieve a sufficiently controlled condition to be able to derive brain activity. What we are starting to do, what we are starting to do in Parma during my ERC and in many other laboratories all over the world, is to apply newly developed wireless technology in order to be able to pick up neural activity during active and freely movement of the animal model. In this case, you can see a rat, which is supported by a system because the rat is paralyzed because of a spinal cord lesion, and by deriving neural activity with the same system, with the same way that you have seen applied to human a few minutes ago, it has been possible for research of the Gregoire-Courtine group to decode neural activity and to use the population and multi-unit activity to generate an electrical stimulation pattern to be delivered directly to the spinal cord in order to restore the pattern of movement which is typical of walking. In this case, the system to be controlled is much more simple than the end. Of course, the end is one of the much more challenging organ to be motorically controlled of the human body. It's easiest to restore walking because there are these central pattern generators that, if properly activated, also in terms of their proprioceptive component, can essentially generate by themselves the motor pattern that can enable the animal to walk. After having validated this kind of approach in rats, they moved to non-human primates, and as you can see here, they have been able to use this population dynamic approach to decode the neural activity pattern correlated with walking, and then after producing a spinal lesion to the monkey, they have been able to use this neurally decoded activity in order to trigger temporally specific and very precise electrical stimulation of the spinal cord beyond the lesion in order to reproduce and to allow the animal to walk again after the spinal lesion. And I try to reduce the volume. What they have been now able to do is to apply these results obtained in non-human primate to make, if it works, otherwise you have to believe me. Okay, this is a paralyzed patient with a partial lesion of the spinal cord, which it's extremely difficult to walk, of course. Here you can see that muscle activity is basically silent, and here with electrical microstimulation of the spinal cord, temporarily triggered by the walking pattern, it can actually be able to walk in a pretty good way. And what they have been observing now is that after a long period of neurorehabilitation combined with the stimulation protocol, some of the patients, now they have implanted 6 patients, are able to start walking even without the use of the electrical stimulation. So, to conclude, from a beginning in which typical and conventional neurophysiology has been the core for the discovery of the basic properties of the motor system, by adding neuroanatomical mapping, tracing studies that enable to understand the network, and an increasing amount of computer science, engineering, and physics, it has been possible to decode the way in which our brain controls our movement, the way in which it represents action, to shift the focus from the concept of representation at the single neuron level to system dynamics. And in this way, it is more and more possible to exploit this information for translation application. On the other side of the basic research, we still lack the possibility to investigate the system while it is working in close to natural conditions. And so ethology, and in particular neuroethology, is necessary in order to crack the code of brain functioning during daily life situation. And this is what my lab in particular is much more involved in this last year. And I hope that I will have something more to say in the next year. So, thank you very much for your attention.